All right, I'm going to start by showing how to do the chi-square test or how to use this chart here. All right, so I'm going to come back to this table in a bit. Right. Let me explain how to use it and then do a quick example. So this will come under genetics. So when you're doing experiments, so if you remember with like the dihybrid cross, right? If you cross two heterozygotes offsprings in terms of phenotypic ratio, you expect a 9 to 3 to 3 to 1. So based on Mendel's laws, right? If you do a certain cross, you will have a certain expectations of the outcome. The purpose of doing a chi-square test, you will have your expectations, and then when you do an experiment, you're going to get some results. What you want to know if those results that you get, if the difference, so what will you say? The difference between our observed, there will be no difference between our observations and our expectations. But if there is any difference, it will be due to chance. So this table here and our chi-squared value, we are going to determine if any differences between our observed results and our expectations is due to chance. All right? So that is the purpose of this. When we do a cross, we have an expectations. We're going to get results. We want to know if it is due to chance or not. All right? How do we use this table? The first column here is the degree of freedom. So you have 1, 2, 3, 4. I will show you how to get the degree of freedom. Just a second. All right, so this is their degree of freedom. For biology, these readings up here, the probabilities, we use 0.5. Sorry, this is 0.05. So if this should come on the exam, do not worry about these other readings. So we are interested in degrees of freedom and this column here with the probability 0.05. So what you're going to do, let us say your degree of freedom is 2. You're going to find where it intersects the 0.05. So you have a degree of freedom of 2 and you have your probability of 0.5. You're going to look at this value here. And based on what we calculated, we are going to see if it is greater than the 5.99 or less. So this number that intersects with where the degree of freedom and the probability meets, that is our critical value. All right. So you find your degree of freedom and you match it with the 0.05. So let us do a quick example. By the way, before I move, any question? All right.
Yes, sir. So the degree, the way board gone.
Me clear it already.
All right, no bother.
Yeah, continue.
So the degree of freedom for the two would that be 5.59?
Right. So we're going to use this critical value of 5.99.
Okay.
Yeah. All right. So let's show you how it works. Go back to the whiteboard. All right. So everybody is supposed to know the experiment with Mendel and the pea plant, right? And what are the phenotypes with the dihybrid cross with the pea plant? Ronan yellow, wrinkled green, that experiment. One second to just connect this. All right, I'm just going to use the, all right, so which one is this? Do you know how to do a null hypothesis?
No, sir.
All right, so your null hypothesis with, all right, hold on. So let us say you have a plant, right? Let's use B for blue flowers. Move quickly. And small b for white flowers. Right? And we cross two heterozygous offsprings. So big B, little b, big B, little b. So in this box it would be big B, this box, big B, little b, big B, little b. What is the phenotypic ratio? How many blue flowers will we have?
3 blue flowers to 1 white.
Okay. Yes, so the phenotypic ratio is 3 to 1. What would be the genotypic ratio? You start with your homozygous dominant one. So how many would be homozygous dominant?
1. Wait.
1. Then you do the heterozygous. How many would be heterozygous?
2. 2.
Right. And how many would be homozygous recessive?
1.
Yeah. So for the chi-square test and null hypothesis that we're going to do, we are not interested in the genotypic ratio. We are interested in the phenotypic ratio. So what is this phenotypic ratio saying? If we have 4, let's start out with, if you have 4 flowers, right? Out of the 4, how many should be blue?
3 would be blue, 1 will be white.
Out of 4 flowers, 3 blue and 1 white. In terms of percentage, what percentage should be blue?
So you're going to say 3 over 4 times.
75.
Right. So 3 over 4, which is equal to 75%. So 75% of the flowers should be blue. So whenever you do a heterozygous cross for monohybrid, 75% should have the dominant trait, and 25% should have the recessive trait, which is white. So 25%.
When you do a monohybrid cross, 75% dominant, 25% recessive, that's what you said?
For a two heterozygous cross. There's not any cross, it has to be 2 heterozygous offsprings. well, will be offspring parents. So when you cross two heterozygous parents, for the phenotype, 75% should be of the dominant trait, and 25% is of the recessive trait. So question, if you have 50 flowers, right? How many should be blue? So if you cross the offsprings, two heterozygous ones, right? Not offspring, parents. And you get 50 offsprings. How many of it should be blue and how many should be white?
49 blue, 1 white.
No, man. Look at the
Wait.
percentage.
It will be 12.5.
So you get 12.5 for the
What are
white?
the 50?
Yeah.
Yeah, it would have been 12.5 and
All right, so 37 blue and 12. But run it off to 13 would be white.
So I don't round off the blue too because it's 37.5.
Well, I one would have run off. Can I go have half of?
Oh, all right, yeah.
Over there. So if we're doing an experiment, right? Before we do the experiment, 37 of the flowers is expected to be blue. So this is our expectations. 37 would be blue and 13 will be white. All right, that is our expectation. Let us say we do the experiment now. And you get, let us say you get 25, not 25. Let's say you get 31, that is white, and 19, because it's 50 in total, 19, that is 31 blue and 19 white. So before we start our experiment, we would have a hypothesis, null hypothesis. What we're going to say, so essentially you're saying there will be no significant difference between observed and the expected. So there will be no significant difference between observed and expected. So at the end of the experiment, we are going to either accept the null hypothesis or reject it. So how are we going to do that? We have to do a little calculation. So I need to clear some of the word. Give me a second. All right, yeah, sorry about that. All right, so, right, the chi-squared value, if we want to work it out, this symbol here, anybody know what this symbol means?
Sum of.
All right, observed minus. So O is for observed, E is for expected. All right, divided by E. All right, in the interest of time, I'm just going to use some data from this study guide. All right, so, all right, let's put my rational. All right, so I should probably screenshot it and put it. Give me a second. Let me just. Give me a second. Let's do this. I'm just emailing the page so I can bring it up on this. Zoom out a bit. All right, ready again. All right, so everybody can see the table now, right?
Yes, sir.
All right, so this is the formula. So that means sum of observed minus expected. So this one is a dihybrid cross. Right, so for in this particular example, right, they were describing tomato plants. So remember, in dihybrid cross, we're doing two types of traits. So in this one, it was purple cut. Okay, so these are the two traits. Their expectation was a 1-to-1-to-1 ratio. And their null hypothesis, you're always going to say there is no significant difference between the observed and the expected. Yes, I'm going to do genetic engineering shortly. What you're going to do, based on the data provided, you are going to work out the observed minus expected. So here you have the category. The observed was 70. The expected was 81. So if it's a 1-to-1-to-1-to-1 ratio, then they all have to be the same. So the total amount of plants is 324. And if it is a 1-to-1-to-1-to-1 ratio, they all have to be the same. In the monohybrid cross we did, with the 3-to-1 ratio, so apply ratio, one number you get. So in this case, it was 324 plants. So since the ratio is 1-to-1-to-1, everything has to be equal. This is our expectation. Now, for the first plant, this is what we observed. as it says, observed minus expected, you square it. So it would be 70, take away 81, which you get minus 11. And then you square it, you get 121. You divide it by the expected, and this is the final answer. So you would do this for each of the categories. And when you get all the answers, remember this E, kind of like an E, means sum of. We're going to add up the 0.2, the 0.3, 1.23, 1.49. And that is your chi-squared value. All right, so that is what you will do. Now, once you get this value, we're going to go to the chart I was showing you earlier. But before you go to the chart, you have what we call degree of freedom. Well, I told you about it earlier, right? So the degree of freedom. So we have 1, 2, 3, 4 categories. So I'm just going to call it n minus 1. So n is the amount of categories. And once you get, if you get a dihybrid cross, the number of category is going to be 4. So once it's dihybrid, you will have 4 categories because you will have 4 set of phenotypes. 4 minus 1, that will be equal to 3. So this is how you get your degree of freedom. Is that clear? Oh, let me go back for the answer. So the answer was 3.23. So the first thing, what is the first thing that we should do now when we come to this table? Anybody? Which column should I pick?
Oh, look for the degree of freedom, which is 3.
Third degree of freedom.
Right, so 3, and then I go across to where?
Go across to the
It is 0.05.
Right. So this number here, right?
Yeah.
All right. So now what we're going to do is compare our answer, which was what, 3.23? I think it is 3 point something. Once your answer that you calculated is less than the critical value, you're going to accept your null hypothesis, which means that the difference between our observed and expected, it is purely due to chance. All right. So once
repeat what you said about once the calculated answer is less than the what?
the critical value. So this 7.82, that is our critical value. So you compare your chi-squared value that you calculated to the critical value. Once it is less, you accept the null hypothesis. So remember, this calculation is to determine if our you did your experiment, right? What you're trying to figure out, this difference, you expect to get 81 purple cut plants, right? And you got 70. Was it due to chance? Or was there something interfering with the process? So your null hypothesis is saying that any difference that you would see is due to chance. So once it is due to chance, it is not significant. So when you do your calculation, you come to this table. So this is the table that you are going to use to tell you if the difference that you got, it is significant or not. So the critical value is 7.82. What we calculated was 3.23. The 3.23 is less than the 7.82. Therefore, we are not going to reject the null hypothesis. We are going to accept, which means that the difference is due to chance. Is that clear? So once it is less, it is due to chance. If it exceeds, yeah.
So the reason why that third Third one because the value we get at 3.23.
No, the degree of freedom. So
Oh.
to get the degree of freedom, it is the number of categories, so it's n minus 1, which is the number of categories, which is 4. So once you do a dihybrid cross, you're going to have 4 categories, and you take away 1 from it, which leaves you with 3. So so the degree of freedom is 3. So that is why you come to this here. And we are for biology, we're using 0.05. So look where the 0.05 and the degree of freedom meet. This value, yeah.
Them that give one table like that's something that we say observed minus expected and we put in those values.
No, I more than no, I would work it out.
We
Well,
don't work
they,
that out.
yeah, they
Okay.
can give about more than likely they will want you to work it out.
Okay.
Or at least some of it, yeah. So ensure you remember the formula.
Sir, question.
Yeah.
Sir, what if they leave like the expected value come blank? How would you work that out? It's so nice. Sir, what if they left like the expected results column blank? How
It
would you
will
work
be-
it out?
Right, it will be blank. All right, so right, the expected, as I was telling you, is based on your not. So in the first part, let me go back to the, it's still on the board. Just a, let me stop. Just a second. Right, so remember, when you cross 2 heterozygous offsprings, right, for a monohybrid cross, what is the expected phenotypic ratio? Right, that is where the answer comes from, you know. So when you do a monohybrid cross for 2 heterozygous offsprings, what is the expected phenotypic ratio?
3 to 1.
Right. So so once you do a heterozygous cross, it is 3 to 1. So this is what you expect. Now, what you're going to do is apply this ratio to a particular number you get. So I told you it was 100 flowers, right? So if you were doing the cross with, or if you get 100 offsprings, of the 100, how many will be blue? Or how many do we
75.
expect? Right, so we expect 25 to be blue. And
Also,
how much do we?
the rest 30 something. 25.
Right. So the expected results is based on the type of cross we are doing. So they don't need to give you the expected results. You are supposed to know it. So you can work it out.
So we have to do the monohybrid cross and all of them stuff there.
Yeah, and if it's a dihybrid cross, you would have to do it.
Okay.
So, right. So for the dihybrid cross, right? If it's 2 heterozygous offsprings, instead of the 3 to 1, so 3 to 1 that's monohybrid. But the dihybrid is 2 traits. So the phenotypic ratio, let me just clear it.
Sir, before you talk about that, what do you say, like, however, if the calculated value is greater than the critical value, what will happen?
You reject the null hypothesis.
Okay.
Everybody familiar with Mendel experiment, right? And the green and the wrinkled pea plant? I'm assuming that, all right? So for a dihybrid cross, heterozygous. All right, so 2 heterozygous plant. You will get a phenotypic ratio of 9 to 3 to 3 to 1. If you're not sure, let me just remind you. So big R is round, small r is wrinkled. All right, that is in terms of the shape. In terms of the wrinkled and green, yeah. Big Y is for yellow and small y is for green. So in terms of the phenotype, the nine of them would be round and yellow. Round and yellow, it would be 9. Then if round and yellow, which other trait can you get?
Round and wrinkled.
All right. Round and wrinkled. 3. Which other trait I can get?
Yellow and green.
All right. Yellow and green. Which other one?
Wrinkled and green.
Sir, around and green, not yellow.
Thanks. Okay. Wrinkled and green is 1. What was the correction?
Brown and green.
Round. Oh.
For the third one. No. For the one
The
below
third
that.
one. You have 2
Oh.
colors for the third one. Can you
Alright.
erase them?
No, but we can't have round and wrinkled because round and wrinkled a shape.
Sir,
So-
yeah, that one fixed too.
Right. Yes.
That brown and green.
Right. It's supposed to be shape with color.
So,
So we have
round
round
and
and
yellow.
Oh,
yellow.
round and green and
Right.
yellow and wrinkled.
So round and green.
Green and yellow and wrinkled.
All right. All right. So round and yellow, that's 9. So no, it don't have to be people. I'm just going to tell you no. If you get a dihybrid cross, no matter which 2 traits that you are combining, right? The big R that is dominant, big Y that is dominant. The 2 dominant traits that will be the 9. All right. The 2 dominant traits that will make up the 9 in your 9 to 3 to 3 to 1 ratio. The homozygous recessive one. So look, wrinkled and green. Those are the 2 recessive ones, right? So the 2 recessive ones, where when they combine, you are going to get 1 in terms of the ratio. That's the 1. And then no, dominant with recessive, you get 3. Let me put that on the board quick. It can help you if it comes. So dominant and dominant, right? That is 9. Dominant and recessive, 3. Recessive and dominant, 3. Recessive and recessive, 1. All right, so this is how the phenotypic ratio will work for a dihybrid cross. Now, when you get, all right, so let's do it quickly. If you have 5 on, all right, let's say I have 375 pea plant, right? How much do you expect?
Excuse me, sir. So you're writing this now?
Say that again. If I'm?
Are you writing this now, whatever you're saying?
Yeah.
Because it's still on the null hypothesis page.
Yeah, I'm on the null hypothesis page. Hold on, all right. Tell me if.
Sir, we're not seeing what you're writing then.
Yeah, that's what I'm saying.
All right. I'm going to. I just erased the word hypothesis. Are you still seeing it?
No, yeah, we're seeing it, sir.
All right. Let me stop share and click it back. Are you seeing it now?
Yes, sir.
All right. Yes, yes, just. All right, so in terms of doing this on the exam, the question will come, they will give you a cross to the right. Once you do the cross, you will get your expected phenotypic ratio. So they this one. So what I'm saying now, in our example here, or in any example, once it's two heterozygous offsprings, your phenotypic ratio is 9 to 3 to 3 to 1. What I'm showing you, to save time on the exam, the two dominant traits, that will be 9. In our example, it is round and yellow pea plant. The dominant and recessive, meaning round and green, yellow and wrinkled. Dominant with recessive, that will be 3 and 3. And recessive with recessive, that will be 1. That is how the ratio works. So you won't need to be counting up in your table. Is that clear? You don't need to count up your table. So now, my question now, if you have 300 if you've got 375 pea plant, how much of them do you expect to be round and yellow? What would we do? How would we get the answer?
Sir, 57 of them. Wait.
Tell me the calculation you do.
Sir, you said 3. Wait, on the percentage?
375 plants, 375 pea plant. How much of them will be round and yellow? Right, in the interest of time, it's 9/16.
It would have been
So-
19% to be round and yellow.
So 9/16 times 375, how much I get?
Yes.
111.
How much?
Sir, 211.
And for the round and green, it would be 3/16 times 375. How much I get?
70.
All right. $70. And then it would be the same for the, all right. So this will get 211, 70, 70. That would mean this one is how much?
23.
23.
All right. So this is what we expect, right? I'm going to make up some numbers quickly. This is the made up number in block. 150. Somebody will work out observed minus expected squared quickly for me for this one. Divided by expected. All right, and tell me the answer. And next person, work out this one quickly. 150.
So the observed value would have been the one that will calculate, which is 211.
No, observed is in block. Expected is what you calculate.
Okay.
Yeah.
Sir, the first one is 61.
Give me a second. The first one I get 61. You divide it by the expected number. So you do
Oh no, I'm not do that yet.
Alright. So you must do 150 take away 211 squared
So I'm gonna get
divided by
17 point. Oh wait, I don't know. Wait.
each person just work out one of these for me.
So for the first one, I'm gonna get 17.6.
All right, thank you.
Second one, I get 1.25. I'm not sure though.
For the second one, if you did
For the second one is 1.42.
1.42,
1.43.
for the third one,
No, sir, it's have to be 3 because the 8 come after the 2.
Sir, I'm gonna get 1.25 for the second one. And a 70 minus 80.
1.25.
no,
1.25.
80 minus 70, observed
Sir.
minus expected,
Sir, you just say the observed one.
in black and the expected in green.
So number 3 is 0.
Right. And number 4,
Sir,
36,
and 17.57.
please repeat, how much?
117.57.
Anybody else? Just to double check.
Yes, sir, 117.57.
That sounds big, all right. 117.6, all right. All right, add them up now for me quickly. How much was it? 117 point, what you said, 57?
Yes, sir.
All right, so just add up the 3 of them now.
Sir, why you put 0? Oh, no bother.
All right.
$136.6.
Yes, sir.
All right. So our value is how much? 100. Sorry, repeat that again.
136.6.
36.6.
All right. So now, and our degree of freedom is how much?
3.
All right.
3.
Let's go to our chart now. Tell me what to do,
Look for your degree of freedom.
which is,
3.
all right. Oh, on the wrong chart. Right. Where is?
So
And then
because
go.
it's greater than it, it's greater than the 7.82. It
All right, so
wrong.
we locate the 0.05 and we locate the 3, right? And it meet here. So what would we accept the null hypothesis or would we reject it?
Sir, it reject it because the calculation
Reject
of
it.
7.82.
Yeah.
Right, greater. So the difference between observed and expected, let me just go back to it now. Remember, our null hypothesis says that there is no significant difference between observed and expected. What is our chi-squared value telling us? Is it saying the difference is significant or not significant?
Not significant.
30 significant.
Right. Remember, once it's over it, it is significant.
Sir, repeat the
So-
question.
The search said significant.
So this value is 136, right? Our calculated answer.
Yeah.
Right. What is the critical value from the table? 7 point something, right?
Yes.
So once this value is greater than the critical value, we reject the null hypothesis. The difference we get, it is significant. We clear on that? The answers in green is what we expect to get. Ones in black are what we observed. When we did our calculation, our chi-squared value is 136. When we go onto the sheet, clearly, you find your degree of freedom, which is 3. You go across to 0.05, it meets at 7.82. 100 and whatever we just got is very, very larger than 7.82. Even if it was 7.83, once it is above it, what do? We reject the null hypothesis. The difference was not due to chance.
So, sir,
Mhm.
once the null hypothesis is rejected, that means that the significance is correct or something like that, whatever.
No, hold on. Before you go to the null hypothesis, this is what you are using to accept it or reject it. We calculated our value to be 136, right? Is bigger than 7.82. Once it is bigger, you reject your null hypothesis. That's simple. All right?
Yes,
Clear
sir.
enough? Yeah. And what I'm saying, it was clearly, definitely not due to chance, because I mean, just make up some numbers, right? As you saw. So it was not due to chance, and me make it up. So that is why the difference, it was significant. Because look, if you expect this to be 23, and we put 75, that was some significant difference. Do you understand how it works?
Yeah, I understand now.
Right, it wasn't due to chance. I interfered with it, yeah. So if I did an actual experiment, even if there's a difference, it should be due to chance, all right? So this is, so what now? After you do the null hypothesis, you would come with an alternate, which is basically a rejection of whatever the null hypothesis is saying. So the null hypothesis is saying that there is no significant difference, but clearly there is a significant difference, right? So that is how we use this and apply it. All right, we have to move on. I'm going to stop sharing now. But I'm going to clear the screen. Which one do I want to do next? Let's do some genetic engineering quickly. In genetic engineering, what are we trying to accomplish?
Decide on both favor or something.
Hmm? All right, so as it says, engineering and genetic. So we're making some modifications, right? And so let me just outline the process. And the eggs, using insulin as the example. All right, so as we all know, we need insulin, but the condition with the diabetes, right? So everybody produce it, but diabetics, depending on the type, they will need their insulin. So before bacteria, well, this is a crash course, so no history. Let's just get straight to the procedure. So we need to make insulin. So the first thing that we need to do in genetic engineering, what we're doing, we're taking a gene, right? And remember, a gene is just a specific portion of DNA. So we are taking a gene and put it in a vector. So gene goes into vector, vector goes into host, and the host produce the protein. From the gene. So remember, the gene codes for a protein that will be expressed. So you put the gene into a vector, you put the vector into a host, and the host will express the protein. So how does that work? Where does the gene come from? So step one, you need to isolate the gene. Right? After you isolate the gene, this is the basic step. Isolate the gene, place the gene in a vector. Place the vector in a host. All right, so the vector is a plasmid, typically a plasmid. And as I know, plasmid, where do we find plasmid?
Bacteria are prokaryotes.
Right. So the host is the bacteria. All right, so we need a little diagram now to outline this. So you just isolate the gene, place it in a plasmid, plasmid in bacteria. Anybody writing? I want to clear the screen.
Sir, you isolate the gene.
Mhm.
What is the vector? The vector is the bacteria,
No, the plasmid.
and then you put the plasmid into the
Bacteria.
vector. Oh,
I'm going
okay.
to show it now via a diagram. So you have your bacteria. Remember now, the bacteria, it will have the chromosomal DNA, right? But we're not interested in the chromosomal DNA. Inside of the bacteria, you have a circular strand of DNA. That is the plasmid. So the plasmid is DNA. So a circular strand of DNA, that is what we call the plasmid. It is inside of your bacteria. All right. So we have our technique to basically punch holes into the bacteria to get out the plasmid. So we remove plasmid from bacteria. All right. And the plasmid is our vector. So once you isolate your vector, we also need to isolate the insulin gene. So in this case, we isolate the insulin gene from the pancreas. All right. No, this is in the form of mRNA. But what we actually need is DNA, right? The messenger RNA, we convert it to DNA. And the enzymes that does this is called reverse transcriptase. So enzymes that convert RNA into DNA, we call them reverse transcriptase. So the HIV virus of that enzyme, reverse transcriptase. We call the DNA complementary DNA. So if you see cDNA, right, copy DNA or complementary DNA. So you convert the messenger RNA into DNA. You have your plasmid. Now we need to fuse the plasmid with the DNA. I am using this to represent the DNA. All right. And this is my plasmid. Right? So what do we need to do? There's a specific class of enzymes that we use when we're doing genetic engineering. Anybody know the name of the enzyme or the class of
Are
enzymes?
there restricted enzymes then?
Right, restriction enzymes. This process cannot be done without restriction enzymes. Two types of enzymes. The restriction enzyme, which is for cutting, and DNA ligase, which is for joining. So it's like cut and paste. The restriction enzyme is the scissors, and the DNA ligase is the glue. So what we need to do now is cut the plasmid.
Sir, can you repeat the enzyme, please?
Here. So the general name is restriction enzymes. So we're going to cut out a portion of the plasmid. So there you have it. So this is now recombinant DNA. Reason why I call it recombinant DNA, it's DNA from two different organisms. This was from the bacteria and this is from human. All right. And the reason why it's the vector is what does a vector do? So like mosquito is a vector, right?
Carry the gene.
Right. In this case, right, it is carrying something. So the plasmid is carrying the gene. So the plasmid, even though we call it recombinant DNA at this point, so this is our plasmid vector because it has in the DNA. The isolated plasmid, isolated DNA, cut the plasmid, insert the DNA into the plasmid. Once you do that, you place it back into the bacteria. So the bacteria will have its own DNA and then we put the plasmid back inside as well. So we have our technique to punch holes into the bacteria and allow it to take up the plasmid. All right. So let me just annotate this. Insert plasmid into bacteria. So before you go into the specific, just remember what is the general, the three main things in genetic engineering what are the four main steps?
You isolate the genes, then you place it in a
Isolate the
vector,
gene, place it in a vector, yeah,
and place the
and
vector in a
Place
host,
the vector in the host.
right?
and then
And
put the
once,
host.
right, in a growth medium. So the host is the bacteria, right? The bacteria must be able to replicate itself. So in order to do that, we're going to now put the bacteria in growth medium so it can replicate. All right, so the final step, we just put the bacteria in nutrient media. So I'm going to erase up here. Right, so this would be our, just imagine a petri dish, right? Your nutrient media. Now I'm going to draw the bacteria much smaller now. You don't have to do this part, but so just imagine that these would be the bacteria in the nutrient media, all right? So they will replicate. Once they replicate, every bacteria will have the plasmid inside of it. All right, so all of these will have the plasmid. Let me just put blue, 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 and red. So that is how, and bacteria, they multiply quickly. So in note, like within 24 to 48 hours, you can have a lot of bacteria. And all of them will have in the plasmid with the insulin. All right. And they have their technique as well to collect the insulin. So the bacteria, it is going to make insulin. It has the insulin gene, which means it has the instruction to produce it. All right. And this is the general procedure of a gene that we're interested in. We take it out and put it in a vector, put the vector into the host. And that will now give the host the ability to do whatever it needs to. Right? So like there was tomato to survive the cold, they took a gene from the fish and put it in the tomato. So genes is a code that gives you ability to do certain things. So by putting in the insulin gene, it gives it the ability to make insulin. Right? Can I clear the screen?
Wait, sir, let me screenshot this. All right.
Yeah. All right. If this should come up and they ask you for examples of restriction enzyme, I'm going to give it the name of 3. Example of restriction enzyme. look at the specific type of cut it does and tell you quickly.
So what them were there both ind and BAM?
No, a 3. BAM is called BAM H1 and HIND 3. So they are examples of restriction enzymes. So just remember the name. They make a particular type of cut. Let me just check right. Which one does the 6? Give me a second. All right, so you have a next one. It's called A3. No, these restriction enzymes, they cut the DNA in 2 ways. So for example, A3, it will cut the DNA strand, just cut it in 2, not vertically, but well, you can see it on the board, right? This is called a blunt cut. All right, so just cut the DNA in 2. All right, there's a blunt cut. You have a next cut, it's called staggered cut. So just note 2 enzymes and type of cut. They have blunt cut and they have staggered cut. With the staggered cut now, it won't go straight through the DNA strand like the blunt cut. So for like BAM H1, let me see if it was BAM H1 that did this. Now HIND 3 will do this. So this is how it will cut the DNA, and it leaves it with sticky ends. So we call it sticky ends. All right. So you have 2 types of cut, blunt cut, and you have staggered cuts. When you have a staggered cut, we say it leaves the DNA with sticky ends, meaning, right, so you would have A bonded to T, and T, T, C, G, A. Right, so notice all of this is gone. So these ends, it would be free. All right, so we call it sticky ends. So know a few restriction enzymes, and now you know the type of blunt and staggered. All right. We're going to go to next. All right, we're going to move from there now. Let's get into some variation. Right. What are the two types of variation?
You have continuous and discontinuous. Down.
We have two types, continuous and discontinuous. Right. Now, discontinuous, they're like discrete variations. So with this one, it's like either you have a certain trait or you don't. For example, blood group. Tongue roll. So either you roll your tongue or you can't. You have a specific blood group. With the continuous variation now, you can get a range out of it. So the continuous variation is the one that has a range. So like for height, foot size, right? Anything that can give you a range, which means that, go ahead.
Fingerprint, would that be continuous too?
No, tell me why it wouldn't be continuous. Fingerprint would be, would more than one person have the same fingerprint?
No. So that
No.
is continuous.
Right.
But it can be in the same category. Like you have,
All right, could you plot a histogram for fingerprint?
no.
All right. So if you can plot a histogram or a bar graph, you would put that under continuous variation. And right, so if you're doing a tally, well, I shouldn't say it like that. In terms of, so like with the histogram, it would be for continuous variation. Discontinuous, it would have to be a bar graph. So if you can plot a histogram with the trait, it's continuous variation. If you can plot a histogram, anybody remember how histogram work?
Yeah, you have to have different intervals. The
They
bar
have
they
intervals,
put
right?
together.
And how the bar graph work.
You just have like, you just have
Right.
different categories, and if you're in the
Right,
don't know.
exactly. Just like what we said for discontinuous variation, right? You have a particular category and you're either in it or you're not in it. So quick example, ability to tongue roll, all right? So number of persons, all right? This could be the ability to roll the tongue, all right? So let me just put
Yeah, so you go pie chart.
pie chart, sure.
Yeah.
No, I just want to show that with the discontinuous, you're placing them in distinct categories. So any form of chart that shows distinct category, you can use it. Clear? I don't know. I would say this is thumb rule and no thumb rule, or however. So with discontinuous variation, they are placed in distinct categories. With continuous variation now, so like you would say in terms of feet size, right? So let's say 4 to 6, and you would have that. And then like from 6 to 8, then from 8 to 10, something like that. So with continuous variation, you will have a range. Do you understand? I'm not sure.
Yes, sir.
All right. Now, we need to look at what leads to variation. So if you understand, do you understand the word variation in terms of biology? What does the word variation mean?
All right, the difference in the in a trait of an organism.
All right, within a species, right? So
Yeah.
there will be differences between individuals in a species. It can be due to what? Two main things or two main factors that cause variation.
Meiosis.
No.
Well,
Well,
no, wait.
yeah. All right, genetics and the environment. All right, so so kind of genetic changes that leads to variation. That is the most important one, or the biggest factor. The change it once you have changes in the genome, then you will have variation occurring. All right. Now, if we look at our, all right, let me just clear this. We're looking at what causes variation. Let's put that on the board. So it's environment and it's genetics. When it comes to genetics, it is mutation. We're changing up structures within the DNA. All right. So what causes variation? Environmental factors. For example, what? All right. Let's say I have an identical plant, right? And you put two plants, two identical.
Factor.
Okay, just a second. If you have two identical plants, right? And you place one under ideal conditions. And the other one under harsh conditions. So remember, we start out with two identical plants. What we are doing here is looking at how environmental factors can lead to variation. So if you have two identical plants starting out with, and you put them under different conditions, will they look the same? So like, will they have the same, will they go to the same height, same width in terms of their leaves? What will happen? Will they look the same? That's the question.
No.
No, so
So right, there might be what change variation in height.
Sir, what about color?
Right, so variations may occur in height, color of leaf, the size of the leaf as well, right? size of leaf. Let's say it's a fruit tree, right? The size of the fruit can also be affected, right? Size of leaf, size of fruit.
Sir, and one can bear fruit and maybe one don't bear none.
Probably that too, right? So the question now, the height, the change in the height and the color and the size, right? It would probably be due to the amount of nutrients, water, and so forth. Question, would this impact the gene in the plants? Did some change in the genome occur? Or it's more physiological?
Sir, if it's the identical plants, I don't think that's gonna change. Only like the physical features, right?
Right. So the physical features are changed. Good. So then, if the plants reproduce, right, would that affect the future, the offspring, in terms of the genes the offspring is getting? Is the offspring getting the same gene or different gene?
They're the same gene because it wasn't altered. It's just
Wasn't
like
altered.
right.
Right. That's the point. The gene wasn't altered. All right. It's just physiological changes. So it never grew. If it had gotten the nutrients, it would have grown properly. But the genome was not altered. All right. So what is this suggesting? If the variation is in like height, size of leaf, color, and fruit, right? Could we put that under continuous or discontinuous variation? These features,
It can go under continue.
it would go under continuous. So when the environmental factors causes variation, it is not influencing the genome. So the type of variation it leads to would be continuous. All right. So environmental factors, it's not affecting the genome. So it would not lead to any discrete phenotype being formed. What if now we change the genome? So mutation, what is a mutation?
Will affect the makeup of
The
the
gene.
gene.
Right, once you interfere with it, you cut it, you add it, flip it, do something to it. Once you change it, it mutates, all right? The mutation, it can be a single base. And when you interfere with it, it can have serious implications. All right. So it's not like with the environment where it not have enough water, so it grows short. It can be like sickle cell. One, you interfere with a base, and the shape of the red blood cell, it change. So with mutations, that one is more serious, all right? And it can lead, and that one, it leads to discontinuous variation because you're actually now interfering with the gene. Right, let me just clear this. So we can have mutations. We can have mutations in a gene, which is the main mutation there, point mutation. Anybody notice a point mutation? A point mutation, the change so it's just a single change in a nucleotide. And an example under this, you have sickle cell anemia. It's just one change, and it basically messes up everything. One simple change. All right. So point mutation, a change in a single nucleotide. Now, the chromosome can also have mutation. Anybody know of them? The type of chromosomal mutation, insertion, deletion, remember that?
Oh, yeah.
Translocation,
Translocation, right?
mutation, inversion.
Mutation.
Give me a second. So we can have, repeat them for me.
Insertion,
Translocation,
substitution,
duplication,
Sir, isn't insertion
Point.
gene mutation? Isn't it
On the
inversion? I'm not hearing you.
Yeah, hear me now? Not sure.
Yeah.
Repeat about the insertion if it's a point mutation.
Yeah, I'm asking if insertion wouldn't be gene mutation, and inversion would it be the chromosome, because inversion are when you like switch the position, but insertion are when you put in a whole new one. If you understand what I say.
Yeah. I'm going to do an example of each of them quickly. All right, the names kind of, for some of them, the names will actually give you a little hint. So you can add, you can delete. So if it's delete or add in, you have what we call frame shift. You use the term frame shift before? No? All right, so if you're adding, all right, let us say let me not say ABC. Let's use actual. So AGG CU, right? A. Let us say this is the normal genotype, right? Or the normal sequence of nucleotides in the gene. If it says insertion, what do you think we should do? For insertion. The name explains itself. Insertion. What are we going to insert?
Nucleotide base.
Just right. So this is your normal. The abnormal one would arise from a mutation. So if it's insertion, just put something else in there. So let's say AGG, between the G and the C now, we just add a new letter. And then you continue with CUA. All right? So you're adding a nucleotide for the insertion. What would be deletion?
yeah, delete one.
Exactly. So this one that we just did was insertion. All you're doing is just put something. So for the normal one, the abnormal one, it has something new. Question. All right. This is the normal sequence, right? AGG. If we're making an amino acid, how many bases do we read each time for the amino acid? If you're making a protein, all right. So AGG, that would be one amino, right? So AGG, that would make one amino acid from transcription, translation stuff. The next amino acid would be CUA, right? No. What just happened here? Yeah, you will have your AGG, right? But will you still make the amino acid that CUA codes for?
No.
Right. Since we're talking about amino acid, let me just use a base that is actually in. So AGG, you make amino acid. Will you make the next amino acid CUA codes for? Yes or no?
No.
No, right, because it shift out of frame now. ACU. So looking on, let us say AGG, a glycine, and CUA, a lysine. In this DNA, you're not making lysine. You're going to make a different amino acid. So now you have a different amino acid, which means that whatever is being produced is going to have a different function, which is the case in the sickle cell anemia, because you switch out something, a different protein, a different amino acid is going to be made. All right, so look, just by inserting a different base, so once you change one of them, you're making a different amino acid here, which will cause a problem. Because remember, proteins, the sequence of amino acids in a polypeptide chain, and then it fall upon all that. If you don't get the right amino acid sequence, the protein is going to change, right? So just by adding one base or removing it, the amino acid that is made is going to be different, all right? So let's do deletion. Let's random example, CGAACC, all right? A. All we need to, so this is the normal one. All we need to do is delete something now. Normal, abnormal. So let's go with CGAA. Let me replace. No, I need to delete something. All right, so look at this. So you have GC, CGA, ACC, A. With this one, CGA, ACA. What was deleted?
One of the C.
One of the C. All right. So in these, the name is helping you to know what type of mutation is occurring. But the inversion, as the name suggests, probably not so well. Give me just a second. Let me just. All right, insertion, version. Sorry, go over this, please. Which part? The deletion? Talk quickly. Right, so for deletion, you will have your normal gene, right? You delete something. In this case, I deleted one of the C's. That will cause the abnormal gene, all right? So that is all we're doing, deleting something from the gene. So this is the normal one with the two C's. If you have a gene with one C instead of the CCA, you just have CA. So something is missing. That is with deletion. Right? With insertion, you add something that was not there before. With inversion, this is on the chromosome now. So let's say I have 2 chromosomes. So there's a portion of a chromosome, and next portion is here. Let's say I have A, G, or AC, A. Right, ACG.
This
All
is
you're,
inversion, right?
right, all you're going to do now is to flip it. So ACG, it becomes G up here, C and A, right? So it's just the opposite. So just flip it 180. So ACG, GCA. Give me 1 second.
Sir, we never to get the insertion one.
All right, read it again. What are you saying now, Shaniko?
I didn't to get the insertion one.
Insertion?
Yeah, the insertion.
It just adds something to the
In
normal
the
sequence.
abnormal or the normal?
No, so you have the normal one, right?
Yeah.
For it to become abnormal, you need to do something to the normal one. So if you notice I add an A in red here, so it's no longer the normal one. That's the mutation. But
Okay.
once you do something to the DNA, it's a mutation. All right, so the normal one, AGGCUA. In this one, it having an extra A. That's a mutation. All right. In this one, I remove the C, it delete. That's a mutation. It's not the same as it was before. Down here now on the chromosome of this sequence here, ACG, but on this one, it flipped, GCA. So it's inversion. So on this one, ACG over here, CGA. Clear?
Yes, sir.
All right. For duplication, and then we do translocation.
So the one in red is for inversion.
These two are inversion. It's showing
Yeah.
inversion. Yeah.
All right.
Oh, yeah. Let me leave. Inversion. All right. So for duplication, let us say I have AGGU. Let me just color. We're just focusing on the letters. Right.
Sir, you can maybe bring it up further because I can't really see at the bottom.
Oh, all right. All right. So if it says duplicate, what does duplicate mean?
Make adjustments.
Like, yeah, make a copy, like,
Right.
Make
So
a
you're
copy. Yeah.
right. And you're making an exact, right? And so ACCG, right? So this is the normal one, all right? Abnormal. All right, so we have ACCG. If we duplicate it, you're going to have A, C, C, G again. All right, so that's all. You have 2 copies of it. All right, so that's duplication. Anybody have an idea what translocation mean?
Sir, like one of the letters them that is supposed to be somewhere fixed, it's gonna be like somewhere else.
Right. So we move it and attach it somewhere else. All right, just so this we did was duplication. So with duplication, you're just duplicating a specific portion. Insertion, you're flipping it. Deletion, remove something. Insertion, you add something. Translocation, we're going to remove it and put it somewhere else. All right, so let's see if we can do that here now. All right, so we're doing translocation. All right, so ACC. I'm going to cut out ACC. All right. So this is normal. All right, CCG. So CCG, ACC. So I move the ACC from up here and put it down here. The translocation, you're taking a portion of the gene from one end of the chromosome and putting it somewhere else on the chromosome. All right, so it's just movement. Take it from here and put it at the next position. We understand this?
Yes, sir.
All right. So this was translocation. All right.
Sir, them switch positions, but we're not really seeing the bottom part.
Oh, sorry about that. I forgot. Yeah. Did you see the normal one in green, the ACC? Oh, can I see this one here? Are you seeing it?
Yes, sir.
All right. So, right. So the ACC in the abnormal one, just put it in front of this or behind it. Doesn't matter. Just move it from here and put it somewhere else.
Okay.
Yeah. So translocation, it just means take it from where it is and put it somewhere else. Do you know how Down syndrome come about?
Or extra something.
Hmm?
I like when I'm about extra something.
Yeah, extra chromosome,
Chromosome. Yeah.
right? All right. We are going to that part now. Sir, we need to reach. You're Gucci, you know. All right. No, we can't do that. All right. All right. So to explain, I want to explain two types, the Down syndrome. To explain Down syndrome, you have a term, it's called non-disjunction. And if you're familiar with this term,
No, sir.
all right, so it will occur in either, it can occur in mitosis and meiosis, all right? So what it will lead to is the offspring having less or more chromosome. So you know that some can have an extra chromosome. All right, so let us show you. Okay. Some people have an extra X or one, like some female, if you have two X, they have one X. Or some persons have two X and a Y. So in some cells, all right, in your gametes, right? How much the gamete, is it haploid or diploid? I shouldn't say it like that. After
haploid.
meiosis, yeah, at the end of meiosis, it is haploid, right?
Haploid, yeah.
Right. So after meiosis 2, you will have two haploid cells, right? So let us say this one is from a female now. So we're looking at, so after meiosis 2, to form the gamete, to form the zygote, we would have fertilization, right? So let us say this is a haploid cell from the female, haploid cell from the male. And the two of them fuse, you would get the diploid offspring, right? So let us check how we come about now, where some cells have 3 and some will have 1. So we're doing meiosis, right? So you know that in meiosis, in the first division of meiosis, do we have haploid cells or diploid cells? After the first division,
Yeah, that one.
right? So meiosis, first the division. So remember it has 2. How many division does mitosis have?
One.
One.
Right.
Oh, it's one. Mitosis.
Yeah. Mitosis have 2. In the first division of mitosis, do we have chromatids in these cells or chromosomes? As in, do we have homologous chromosomes or a single strand of chromosome after the first division of mitosis?
Homologous.
All right, should I have, is it this?
No.
Or is it
So
this?
this is a
If
mitosis and meiosis.
Meiosis after the first division. I'm skipping out metaphase, anaphase, telophase, but straight to cytokinesis where it split up. They start a
Yeah.
yeah. So I'm saying at the end of the first division, should the two cells look like this? That is what I'm asking.
Yeah.
Yes. Because what would have happened between this diagram and this diagram? What would have happened? Which stages are missing?
Metaphase,
Metaphase
anaphase.
All right,
telophase.
just a second. So this is prophase, right? Right, so I was asking what happened between this stage and this stage. Just as synopsis. The chromosomes will align here.
Align at the
Equator, right?
equator.
And then what happens after that?
Then
Them I go start get
Them
pulled
a-
apart.
What pulls them apart?
The microtubule.
The microtubule.
Tubule.
Spindle fiber.
Right, that's
Spindle
what I
fiber.
said.
Sorry. Then what is the wait, alright, never mind.
Yeah, ask what is the spindle fiber made of?
No, another one left.
All right. Right. So between prophase
Sir, what's the difference between microtubule
And spindle fiber. Come and think them are the same.
the camera on here, Shanika.
And it,
No, all right. Microtubules are like the apparatus then that make it. You have your spindle fiber, right? But what is it actually made of? That's the difference. As in, what make it up? But a revision, so we can't go too much into that. Right. What I was expecting you to tell me is quickly that they line up at the equator, the spindle fibers attach at the centromere and pull the whole homologous chromosomes apart. Right, but if you don't remember, let us just do it quickly. So after prophase, what should happen in this cell? What should it look like?
the
It
third
is metaphase.
pull them apart. It will look like a two different cell lines.
it's a going in middle to metaphase.
Oh, I'm
Right.
thinking that's about anaphase, sorry.
No, yeah, metaphase. So the chromosomes would be in the middle of the cell, right? Does the spindles attach it?
No.
No.
Sure. No
Yeah.
attach.
Yeah, them for them and them.
Right,
Yeah, at equator.
right.
Oh, yeah.
So by the time we reach anaphase, what happens now?
They're
Them I
being
get
pulled
pulled
to
to
the
the
end of
opposite
the
poles.
yeah.
Them
Right. And it's a kind of bend, right? So,
yeah, like that. But you would have to have the two of them.
right, I'm gonna try, yeah. I'm gonna do this.
They
Yeah,
just
it's a
try to
bend.
X them.
Yeah. I'm gonna try to, yeah, it bend. All right, that, yeah. All right, yeah, see there. Yeah, something like that. All right, I have the spindle fibers attached. No, the thing is, all right, so just like how this is happening, right? And then we know that these cells are going to do what? They will eventually split in two, right?
Yeah.
Yes, sir.
And then one is up here and one is in here. Non-disjunction means that this did not happen. They did not, for some reason, it not separate. All right. So this would be the normal process, all right, of meiosis 1. So this is meiosis 1. This stage is anaphase. Now, let us look at the non-disjunction. The non-disjunction is that these two chromosomes, they did not separate. What will happen? One of the cell will be empty and one of them will have what? Two chromosomes
them.
inside, right? All right. So this is where the problem will start, because when this cell undergo meiosis 2, what will happen?
No,
I don't want it to-
No, gonna take
Alright.
place.
Right. So if the cell divide, will anything be inside of them?
No, sir.
So
No. Let me see if I can explain everything on this board. All right. So let's say this is from a male, right? So let us focus on this. What I'm explaining here is non-disjunction, where the chromosomes, they did not separate. The diagrams in red is what should happen. This one in blue is non-disjunction. They are in the same cell. They did not separate, all right? So if they did not separate, one of them will be empty. Good? Now, let us take female haploid cell. Let me use a different color. Just follow me here on this. Right? So the female, select it's an egg. So should two things be in there or one? It's an egg.
One.
One, because it haploid, right?
Yeah.
So right, so this combine with this empty one now, because we're the fertilization. You will get what? One chromosome instead of two, right? Pay attention. As
Yes,
in
sir.
you understand?
Wait. If this one, start from right this one.
You have to follow, right? This right here, the cell, the chromosomes did not separate into the two cells. One have two of them, and then the next one have none. So this one without any. If it fuse with an egg cell, remember it don't have any, you know. So if it fuse with it, this person now, the offspring here, is going to be, will it have extra chromosome or fewer chromosome?
Fewer.
Fewer. An internal syndrome, an X chromosome is missing. So we are showing you how a cell can happen to have a chromosome missing. All right, so non-disjunction, it can make a cell have more chromosome or less chromosome. So you see for these now that have none in it, so understand this one where we get the one. You see the other cells here, they should have three. The end result is that one of them will have three and the next one will have one. Try slowly. All right, let me see if we can do that again. Yeah. All right, so before I move on, everybody understand this, right? We get the one and the zero.
Yeah.
Yes,
All
sir.
right.
Yes, sir.
Right, so we're gonna look now how we get three in one cell. All right, so let us focus on this one, which means I'm going to erase this part. All right. So when this cell divide now under the second second meiosis, how much chromosome will be in each of these cells?
Oh.
Two, all right. Remember, after meiosis, two, how many chromosomes should be in each cell?
One.
All right. So look here now. So, right, so which means that these cells, they should be haploid, but it is actually diploid. So look at this now. Let us fuse it. So 3 chromosome is in this. Let's call it the sperm cell, right? So 1 in the egg as it should be. But instead of the male having 1, it actually have how much? Like, sorry, not 3. It has in 2. All right. So when this sperm with 2 fuses with the egg with 1, how much should be in here?
2.
The sperm of 2, the
3.
egg of
Oh,
1.
3. Sorry, 3.
Right. Now, how many should normally be in this?
2.
2.
Right. So 2 is the normal one, but with this junction, sometimes it might have less or it might have more. So in this case, how much it have?
More.
3, right? We call it, if it have 3, it is trisomy. Trisomy. All right? 1 extra chromosome. So Down syndrome have extra chromosome. It's non, so the 2 you need to know about is Down syndrome and you have Turner syndrome. And it is as a result of this junction. Before we move, what is this junction? What is this junction? What is happening?
So disjunction is when a cell can have less or more chromosomes when division occur.
All right. What is the main? That is partly correct. But what is the main feature of this junction? The chromosomes are not doing what? They are not dividing properly. All right. So remember, like with anaphase, they are supposed to separate, right? But if it does not happen, that is non-disjunction. They're supposed to separate, but they did not. So notice what they end up with is one cell containing the 2 chromosomes, right? And the other one, so this is in one instance where I'm showing you the one cell with both copies, which means that the next cell would be empty. And when it divides by a second meiosis, it would be empty. All right. So non-disjunction, it means that the chromosomes did not separate. All right. And because of that, it will lead to one cell having more chromosomes than it should, and the other cell have less chromosomes than it should. Now, to end this, meiosis have 2 stages, right? So sometimes meiosis 1, it can occur properly, all right? So non-disjunction, it can occur in meiosis 1 or meiosis 2. I know there are a lot of diagrams on the board. So what I'm going to do now, where this is non-disjunction in meiosis 1, all right? I'm going to show you that it can happen after meiosis 2. All right. Let me just erase some of this. Is anybody writing just before I erase? Just checking. I'm going straight up to 6. If anybody wonder, because I will have to leave right after. All right. So if you have to eat or something, you have to just wait or something. But yeah, I'm going straight to 6. All right, so meiosis 1, it will end with 2 cells, right? One set of chromosome up here and the next set down here. After meiosis 1, meiosis 2 will be just like mitosis, right? So I'm going to focus now. All right. Hope you can follow with this. All right. All right, so we finished meiosis 1. starting meiosis 2. This cell here. At the end of meiosis 2, what should happen? Basically, how should it end?
It must be haploid.
All right. We must get 2 cells. All right. One here and one there, right? Question. If this does not separate, split in, if this chromosome does not separate like this, how much would be in each cell? All right, don't answer that. This is what is supposed to happen. Good. This chromosome divides and end up with 2 haploid cells. The next option, so again, one cell has both chromosomes. This one have no chromosome. So what will happen after fertilization?
So which one is that?
Down here. So this cell, if it is fertilized, what will happen?
The zygote
No.
are gonna have one less chromosome.
Right. So again, this I'm just showing you that the trisomy situation, it cannot at meiosis 2 and meiosis 1. So this cell is diploid. So if you have a diploid cell fusing with a haploid cell, then you get a cell which is triploid, 3. Right? So this is supposed to give you 2 haploid cells. That is what is normal. But if it should happen that this non-disjunction occurred, meaning that these 2 chromatids did not separate, then it will lead to trisomy again, where this cell, if it fuses with a next cell, like a haploid cell, then it will have 3 chromosomes. All right? That is how you get trisomy, chromosome 21. That is why they call it trisomy 21. All right? So that's Down syndrome, Turner syndrome. Turner syndrome, it has less. So the Turner syndrome would be like if this one, right, Turner syndrome, this plus this. All right? I'm going to call it TS for Turner syndrome. This plus this Down syndrome, the extra chromosome. All right. So this is just to explain how you can get an extra chromosome inside of the cell. All right. So I hope that you understand it. For Down syndrome, it occurs on chromosome the 21st. So remember, we have 23 chromosomes. Trisomy 21. If you see the term, have you seen the term before? Trisomy. I just explained what trisomy means, right? You have a cell with 3 chromosomes inside of it. So if it says trisomy 21, it is referring to chromosome number 21. So it would not have separated.
Two less chromosome.
Please repeat.
So if it named trisomy 21, may ask if that means it have two less chromosome.
No, it have, no, remember trisomy, I want it to have 3 inside of it, right? So it would have an extra one. Monosomy, I want it, so this one up here is monosomy, it have 1 less. So 2 is normal. All right, so this is, right? This is trisomy. Right? So when you hear trisomy 21, it is giving you, if the 21 is referring to the chromosome number. How many chromosomes, how many pairs of chromosome does human have?
How many pairs? Not
Yeah.
23.
Right. So with trisomy, right? How much would we have?
23 times 3.
No.
Oh, 23 plus 3, 26.
No, it's just, hold on, remember 2 is
24.
normal, right?
Oh,
Okay,
it would have been 47 in all.
Right.
Wait.
But in terms of pairs, it would be
24.
right.
24, yeah.
Yeah, 24 pairs. So it's just 1 extra. So the trisomy, a 1 extra chromosome you have. Yeah.
Wait, sir, repeat, please.
All right. So non-disjunction, it can lead to the cell having an extra chromosome or 1 less chromosome. So like with humans, right? We're supposed to have 23 pairs. So trisomy, it increases by 1. So it becomes 24. And monosomy, it will drop it by 1. Because you're losing out on a chromosome.
So monosome would have 22 chromosomes.
Right. Because if
Okay.
you notice, right, if you notice, after the second division, right, one of the cell is empty,
Yes, sir.
right? So when this empty cell fuse with a haploid cell, how much would be in that new cell? If an empty cell fuse with a cell with 1 chromosome, how much would be in the new cell?
One.
Right. And if a cell with 2 chromosomes fuse with a haploid cell, how many would be in that new chromosome?
3.
3.
Right. Yeah. So trisomy go by 1, monosome when it's empty and fuse with a haploid one. So it decrease by 1. Yeah. So if you know, if them asking if you explain that, yeah, just it's simple, never say simple, but just use all that you need to remember, right? Do not allow the cell, do not allow the chromosome to separate. So you're not doing the normal process of meiosis. So when it gets to anaphase, right? What is supposed to happen at anaphase?
They must be pulled to the opposite pole.
Right. So they must separate, right? And what does non-disjunction mean? What does
It
the term
means that
mean?
it
They
not,
don't divide in the correct way.
Right. So you're not going to separate them. So in meiosis, we are separating, in meiosis 1, you are separating the whole homologous pairs of chromosome. Disjunction would mean to separate them. So non-disjunction, we're not separating them. So you would put the 2 of these in 1 cell, and the next one would stay empty. That is if you are showing it at meiosis 1. If you're showing it at meiosis 2, this cell here would separate to give you 2 diploids, right? And then now for meiosis 2, this one of them, it will not separate. So one cell will be diploid and the next one will be empty. All right, that is non-disjunction. It can happen either at meiosis 1 or meiosis 2. Anaphase is the problem, all right? So with anaphase, it should not occur. So if you're showing it in meiosis 1, anaphase should not take place, as anaphase is where they separate. Is that clear? So just remember that if you want to show non-disjunction, do not carry out anaphase. Well, yes, don't carry out anaphase, but that don't mean this. When I say don't carry out anaphase, that don't mean you should not divide the cells. The cell will divide, it's just that the chromosomes will not go to opposite parts of the cell. All
So
right?
just show metaphase alone
No,
and
man.
prophase.
I will show the prophase, the metaphase. With anaphase, the chromosomes to one side. Do not pull them apart.
Wait till you just freeze up.
All right, so I'm gonna say, when we say don't show anaphase, well, yeah, so I guess skip from metaphase, or if you want, even if you're trying to show anaphase, put the 2 chromosomes one end of the cell to show that it is not separating. All right?
Sir, because in an anaphase, it basically I pull them apart evenly.
Right.
And that's
On a non-disjunction, it's not supposed to pull apart. So apart from that, the final thing, remember meiosis has 2 stages. So if it doesn't, if you do anaphase in stage 1, do not do it in stage 2, right? So because meiosis have 2 stages, you have 2 sets of anaphase. So you can show it at stage 2 or stage 1. So if you show it at stage 1, do not show it at stage 2, right? So yeah. Let's look at some variation. We started with on variation. What I want to get to, where was it? Disruptive, stabilizing.
Sir, they always tend to ask about the bacteria and natural selection, the penicillin thing.
There's something I'm getting to know. So in terms of apart from discontinuous and so forth, right? But a natural selection method into. What is disruptive variation? What happens in that? Anybody? Not disruptive variation. I want to look at selection. So I want to look at directional, stabilizing, and disruptive. Do you know how to explain those 3? So I'm moving from variation to selection. Yes, no? Anybody? Know
Yes,
what happens in directional? All right, let me just do it quickly. All right, so what is natural selection? Because that is what we're heading into. Basic. Natural selection. So look, it's basically the environment. So when persons are animals, right? Let's look at animals. They live in a particular environment. The environment undergoes changes, right? Let's say constantly. Or let's say summertime hot, some winter, it's very cold, right? So based on how the organism is structured, some may be suited for summer, some may be suited for winter. based on the traits that they have, those that have the favorable traits will survive. So natural, because it's what the environment, it's nature. And then selection, particular set of traits is being favored. So select natural, nature is doing it. Some set of environmental factors are going to favor a certain set of organisms, all right? So the type of selections, we have directional. And to do these, we need graphs, all right? So we start with directional. Now, whenever you have a bell-shaped curve, right, in any scenario, this out here is one extreme. So this end, that's one extreme. So the bell-shaped curve, it has two extremes. This is an extreme. This is an extreme. All right. And then in here now, you would have the average there. If it was a bell-shaped curve in terms of grade, out here, extreme, it would be extreme, low, extreme, high. Then you have the average in the middle. We're going to apply these bell-shaped curve to disruptive, stabilizing, and directional selection. All right. So directional selection now, based on what is happening in the environment, it is going to favor one extreme. For directional selection, one extreme is favored. And I'm going to use the example with the giraffes, right? Giraffe, FFE, giraffes with long necks, giraffe with long neck, let's say medium and short neck, right? So we said that it is favoring one extreme. So in this example, giraffe, if they have tall necks, right, they can reach high up in the trees, right? If they are short, they cannot reach it. Medium, yeah, to some extent. So remember, it's natural selection, we're looking at evolution as well. So the long neck, the environment is selecting the giraffes with long neck, because the one with short neck will not reach the food. So over time, you will not have any giraffe with short neck. Now, what will the giraffe with long neck do? They will be able to do what? Reproduce, right?
Yes, sir.
Yes, sir.
Now, the long neck, was it the environment that gave the giraffe the long neck?
No.
No.
No, exactly. Which means that when they reproduce, what type of giraffe will be produced?
Long neck.
Long neck.
Exactly. So over time, what will happen to the population of giraffe with long neck?
It's gonna increase.
All right. So that means what will happen to this curve?
It's gonna bend over.
So it's a go shift for favor the
Right.
long neck.
Which means that it now go start out here so again, because this extreme has now become extinct,
60.
right? So probably start from like, let's say here, and the new peak will be at a higher height. So in terms of if down here, up here would have been the height. I mean the y-axis would have been height of the giraffes, right? So notice the peak, it would be, no, this would be the number of giraffes. Out here would be the height.
Sir,
Yeah.
but do you think the extreme they are gonna stay at the same place, but like the peak would have just shift a little and bend?
Yeah, peak crush. Well, remember two extreme we have, you know.
Oh,
So this extreme out here would be the one with short
long.
neck.
Oh, okay.
Yeah. The one out here now would be the one with long neck. On the x-axis, it would be height of neck or length of neck. Length of neck. So the number of giraffe with long neck would increase. Notice again, it would start about here because they don't have any giraffe with short neck. They are extinct. So directional selection, one extreme is favored. So in this example, we're favoring the extreme in the long neck extreme. So if you have to come up with an example, just make up anything in which one extreme can be favored. So it could be, you can use polar bear, no, not necessarily polar bear, let's say 4, in terms of 4, right? So you make up a condition where the time is, it was hot, and then probably something happened, the time get cold, right? Climate change. So the organisms with thick fur would progress or would be able to survive and reproduce. And those with a little bit of fur would die out. So you just need to create your two extremes. Is that clear?
Sir, the speckled moth would have come under directional, right?
Yes. Remember that one there. So just right. So just make an extreme where one extreme is being favored over the next one. All right. So in directional selection, just remember one extreme is being favored. So that would be it. All right. And if I ask to explain, you would explain what happened with the giraffe with long neck can reach food, survive to reproduce. So a key thing here is that the offspring must be able to reproduce. All right. After we done explain this, we just list out Darwin's observation. All right, so that is how you do directional selection. You have stabilizing now. Oh, but this anchor, this is bad for you. All right, stabilizing. For stabilizing now, the kind of like the average is selected. I'm going to put the average. I'm going to put trait. The average trait is selected for, and the two extremes are selected against. All right. So what that means, all right, hold on. Let me just remove this graph here. And if you're doing this, show the before and after graph, all right? So you can draw the graph 2 times or put 2 curves on the same graph. So before I give an example, if we are eliminating the two extremes, right? So we're eliminating out here and we're eliminating out here. you think the curve will look?
So it's a go thin like in the middle.
Right, because the average is being selected. Will it still start out here and end out here?
No.
No,
Right,
sir.
so it will, right, so it probably start like here and go up. All right.
That have to stabilize.
Right. So with the stabilization, with the stabilizing selection, the two extremes are not favorable for them to survive. So for example, let's use climate. Let's say the temperature is 30 or let's say 35 degrees Celsius every day, right? And you have some animals with thick, like really thick. So let's say at that temperature, it too, them coat it insulate them too much. And then you have some with no coat at all. So let's say half of the year, it's really hot, and the next half it is really cold. So the two extremes, you have animal with thick fur, and no fur, right? And then you have medium fur. So we have two times, when the time is really hot and when the time is really cold. And that happens two times for the year. So when the time is really hot, what will happen to the animals with thick fur?
They will suffer.
All right.
Them are
And
go
when the
boil
time,
up.
right, and they don't. When the time is really cold, what will happen to the animals with no fur?
Them are go
It
dead out.
will be
All
extinct.
right.
Cool and die.
Yeah. So you're seeing a stabilization. Any example, once it looks like none of the extreme will survive, that is stabilization. If it seems like something that does not favors neither having thick fur or having no fur, that is stabilizing selection. So you see for the one with the giraffe, right? How could you adjust it to become stabilizing selection? Just to see if you understand. Which one of the next would be favored if it's to be something like
They're stabilized
stabilizing? Yeah,
medium.
which medium, that is correct. So just give me a second. Right. So we would just change this scenario where long neck or short neck is not needed. All right. So this is what you do for stabilizing, directional, and then the final one now is disruptive. With disruptive now, the average is selected against in directional, one extreme. In stabilizing, none of the extreme. In directional, the two extreme is selected for. All right, so that was stabilizing. Let's do the final one now, which is disruptive. Disruptive. I want to tell you, all right, the two extremes by the way, when I put the two extremes are selected for on the exam, don't just put that. So like you would say the environmental changes favors the survival of the organisms with the two extremes, right? So in this case, you would put, if we're discussing fur coat, it would favor the organism with thick fur and no fur, right? So when we say the two extremes are selected for, or just one extreme is selected for, what it means is that the current environmental settings, it is suitable for one extreme, the medium, or the two extreme. Right? So what will happen now? Let me ask you, how do you think the graph will look if the two extremes is selected for?
Because like go up and then go down.
It would go up and then go down, all right? So the height, the peak, it would drop,
The peak with a low.
right? So it is going to dip. So this part, it is going to be high, all right? Because it is selected for. And then it dip because the medium, the average, it's selected against. So just imagine you are in a class in terms of school, either the students are failing or they are doing really well. So there's no in between in this case. So they are doing really poorly or they are doing excellently. So it's like this, all right? So that is what it means. No average. So really good or really bad, really good, all right? So both extremes are selected for. So this graph is disruptive. But nothing, the average is not selected for. So these are the three.
So we don't have two peak then with the disruptive?
That is correct. The two extremes. All right, so natural selection now. Let's go to drugs and pesticide resistance. So bacteria, right? Before I get that, in all of these examples, the basis of natural selection, right, is about survival of the fittest. They like to use that term, survival of the fittest. What does that mean? When it says survival of the fittest, as you know, there is variation among organisms in a particular species, right? So when we say survival of the fittest, the organisms that have the suitable trait to adapt and survive the environment, that is what is natural selection and the survival of the fittest. The survival of the fittest means some organisms in a particular species will have favorable traits to survive the environment and also to reproduce. We clear on that? Want to write it? Are we good?
Yes, I write it. Please.
First for essentially, I'm brain gone.
ESS.
Yeah, Jeff. Right, essentially. just remember, at least just remember this part. Organisms with favorable traits will survive and reproduce, and the ones that don't have it will die out. So, in one sentence, survival of the fittest, organisms with favorable traits survive and reproduce. The ones without it die and become extinct. That is what survival of the fittest essentially means. Is anybody writing?
No, sir.
All right. One second. All right, so natural selection, we also have artificial selection. Oh, before we get there, with the bacteria, right? Clear this. All right, so the same thing. So the reason why, even with like sprays, like I'm just spray like book spray, the antibiotics, they don't work after a while. The reason being some bacteria. So let's say I have some bacteria, right, in a population. And you apply the antibiotic. So these are the bacteria, and the black dot is the antibiotic. Right? They apply to all of them. Some of them are going to die, not all. All right, so the antibiotic, it will destroy some of the bacteria. But some of them actually survive. Now, those that survive, they are going to reproduce, right? No, when they reproduce, what will those bacteria have?
Antibiotic.
So the bacteria that survived the antibiotics, they would have something in them that made them resistant, right?
Yes,
Yes,
sir.
sir.
So when they reproduce,
Sir,
the new bacteria that is produced, will they be susceptible to the bacteria or resistant?
susceptible.
They would be resistant,
Wait,
meaning
Wouldn't it be resistant?
is repeat
Wouldn't it be resistant?
right, because remember now, whatever made them resistant is in their genome. So when they reproduce, they are transferring that whatever made them resistant to the antibiotic, they are passing it on to the next generation.
also,
Remember,
sir,
mhm.
because these, whatever, them bacteria were able to survive the antibiotic. So when
Yeah.
them offsprings are produced, because they are able to survive that, the offspring get the traits from the parents.
Exactly, right.
Okay, that's why them resistant.
That is correct. So
Okay.
that is why now any bacteria that were not resistant would have been destroyed. The only set of bacteria remaining are the ones that are antibiotic resistant. So that antibiotic will now be useless. Understand? So these bacteria that did not die, they are they have antibiotic, they are antibiotic resistant. And so any bacteria that is made, they will be antibiotic resistant. And so what you will have is an entire generation of bacteria that is what?
Resistant.
Right. So this is just like the giraffe with long neck. After a while, you only have giraffe with long neck. So after a time, you only have bacteria that are antibiotic resistant. The same thing with spraying insects and so forth. Something that is they have in terms of the genome, some variation, right, in their genetic material that allows them to survive. Once they survive, what will happen? They are going to do what?
Interbreed and produce more that will survive.
Exactly. So for natural selection and evolution to work, right, for evolution to work, what must happen? The offspring must do what? Apart from surviving, they have to do what?
Interbreed.
Right. So they have to be able to produce more offspring. If they weren't producing more offspring, it wouldn't work.
No, sir, them would just decrease and dead off over
So
time.
they have to, right. Right, let me do this quickly because 6 o'clock now. A minute for go way at all. All right, process of natural selection. I'm just going to add some pointers here. Process of natural selection. Taking this from online. So you must have overproduction of offspring. Remind me to post a link in the chat online. There's a channel that work past paper video. So we just recap here and you can watch the past papers. So before I will post, I'm just going to post the answer to the 2022. So I'm just going to write the answer sometime tonight and send it out. All right, so overproduction of offspring. Individuals within population will have genetic variation. So it's basically of the over, you have to produce a lot of offsprings. Some of them will have the favorable traits. Once they have favorable traits, they survive, right? Produce more offsprings. And the more offsprings that are produced from one generation to the next, it will gradually just increase the amount of offsprings having the favorable traits. All right, I can just spend 5 more minutes. I'm just going to do, if you don't have this, just take a screenshot. All right, so examples of artificial selection. What just happened? I'm just going to have to put the name. I can look it up. I just interbreeding and outbreeding I'm seeing here. I'm going to end it with speciation, which is a process of forming new species. Anybody know how we can form a new species? One way, it's a process of forming new species, that is speciation. Common example is geographic, but the proper name allotropic. Please repeat.
Allopatry.
You're kind of breaking up. Yeah.
So I was asking if it's not allopatry, another term for geo.
Really need to.
Yeah.
Thank you, yeah. Yeah. Yeah, thanks. Right, so allopathic, this arises from geographic isolation. Isolation. Right, so basically with this one, just moving quickly. You have some insects. These are insects here. All right, something happened and they are separated. All right. So you have, let's say, lizard on one side, lizard over here. Plenty food, little food. I'm just moving quickly at this point. All right. So look, the lizard with little food, they would have to develop certain adaptation. All right. So both of them. All right. So this is like a river. All right. So the point is, something is going to separate a population, a species, right? Over time, they develop different adaptations. So till many, many years after, they basically become new species. They have their genome change. So they are completely different or have distinct features that that makes them different from each other. So geographic isolation, it leads to them finding different ways of surviving. So they, over many years, they become different from each other.
So, sir, like for example, with the same river, you split them in a two. You probably put one in a environment where them have more predator and be
Right.
to survive,
Oh, all that too.
be able to survive. Say one lizard develop where it can camouflage itself, something like
That
that.
is correct. Yeah, man, that good too. Yes. Camouflaging is good, right?
So I can repeat back the explanation that I gave you about the survival and then somebody.
So a bunch of lizard or any insect you want to use, they are living in a particular area. And for something happen and it separate them. So for example, a river come about, all right? So they are separated from each other. As Demetra was saying, let us say on one side of the land, predator is there. So the lizard now have to find some way of adapting. So probably them develop a new color to camouflage themselves. The lizard on the other side with no predator, they don't need to have any camouflage. So over time, that they are developing new characteristics and our traits that will make them become completely different species. All right.
Okay, sir.
Yeah.
Thank you.
I can just put the names of the others because if we don't stop now, we're not gonna get any tax to government. All right, so sympatric speciation, sympatric speciation. But this one, so with allopatric, it's geographic speciation, right? With sympatric, it's something that prevents them from or isolate them in a reproductive sense. Right? So let me show you this. All right, I'm going to post a picture in the comment section of how sim portray, not in the comment section, in the community section. And for those in the Zoom, I will send it. So I'm going to send you the picture of how sim portray speciation can arise. Let me send the link in the what's in the chat for the YouTube thing. Where is it? All right, so I'm going to have to end it here. If I can do any later in the night, I will let you know.
All right, sir. Later.
All right.
All right, sir. Thank you. Later.
Yeah, man, later. All right, so for those in the comment section, I sent a link. That person, they work past paper question for bio. They can just click the link and watch it.
Sir, read my message on WhatsApp.
All right.
Sir, go and look at your taxes.
Right. We have to start getting.